now.
Hi guys.
Hi everyone. So let me maybe start from the very beginning because I'm honored today for guiding you for conference during all 2 hours. We're gonna have a nice discussion because for the first hour we're gonna have a sponsorship hour and the partners hour where our judges will also present the ideas on the longevity, and then we'll switch to the other challenges. And for the challenges, you're gonna have a QA session later. But for today, just to give you an overall explanation of everything we got on the table. So please do not think that that's it, because we're gonna have this Q&A session. So to start with, let me introduce myself. I'm Vita. I'm one of the organizers on this hackathon, and I'm extremely pleased to see all of you here because I do believe that we are all here for beat the aging using the AI revolution. And we will also share with you some of our challenges from the past right after a short presentation from me and Alex. Our partner Gero will present you what we had already done on this field. And I do believe that this time we're gonna have many more new solutions, new ideas, new startups. So let us start with a brief overview of Alex, one another co-organizer of the hackathon, and he will briefly explain you why we decided to organize the hackathon like this. So Alex, go ahead.
Thanks a lot, Vita. I hope you can see my presentation. So it's been an overwhelming success, to be honest. We didn't expect so many people to register. Last time I checked, it were 820 registration. So first of all, the team has done a very strong and good work. So a couple of words about me. 12 years in longevity already, and I will be spearheading rapid adoption track on this hackathon, and I am also responsible for the partnerships. First of all, hackathon team are mostly enthusiasts and volunteers, so please treat them with respect. And we are all here to not earn money, not do something. But we are here to fight aging. We want to live long, healthy lives. So let's fight aging together, and please be respectful to team and to other teams. And one small round of applause from me to the team. We've done wonderful work already. Okay, so crash course into aging. Why we actually need to target aging. Many of you already know it, and you will hear it several times from many other people, but basically all of the diseases, or most of the diseases that kill people today, they exponentially rise with age. So the hypothesis in our field is if we target aging, we can actually postpone these diseases almost indefinitely. So basically we trick our biology to not die in the old age. And if you actually think of how many people die each day from aging, it is about 110,000 people each day. Imagine if there is a meteorite that comes tomorrow and hits a reasonably sized city of 100,000 people. Humanity will do anything to stop it. It will instantly find billions of dollars to actually help these people and evacuate the city and move people out of danger. But we don't do anything like that with aging, even though it is the biggest killer of humans as of today. And like a lot of people think billionaires will solve it. There are numerous billionaires, and they are supposed to wanting to live indefinitely, or they're supposed to spend all of their net worth to fight aging. But in fact, our friends from Longevity Biotech Fellowship did calculations, and about only 0.01% of billionaire net worth is allocated to actual aging research. And if you want to know the best billionaire, I think the best one is Christian Angermayer. And according to my assessment, it's about 3 to 7% of his net worth is spent to fight aging. Also, there are only 10,000 people in the whole world who actually work on solving aging. Only 10,000 people. It's a reasonably sized conference. Yeah, you've probably been at larger conferences, or a soccer or football stadium is usually bigger than that. So there are so few people in the world who are actually doing that we decided, what if we build an army of AI agents in order to assist them? So that's the main purpose of this hackathon. We split the hackathon to 2 tracks. One we called fundamental track. It's a track with well-defined, well-curated challenges. So it's usually for those who want to get a good, well-described task and just do the good work. It can result in a job, collaboration with a big company, or a scientific paper. Rapid adoption track, which I spearhead, and by the way, thanks for the sponsor BIO and VitaDAO who sponsored this track, is for people who prefer more freedom, who probably want to found a startup or earn some money on this agent they built in other ways, or just launch something and see what happens in the real world, but they don't really like any challenges that are in the fundamental track. I will be speaking more about this track in about an hour or hour and a half. Another big thanks to our computational sponsor, Nibius. They will provide each team computational credits, and if a team runs out of credits, you will be able to ask for additional credits from Nibius in the Discord. So we will provide you with the ways to do that. You can use your own computation. We don't really restrict it, but we welcome everyone to test their AI studio. And yes, prizes. Everyone was waiting how how they will earn prizes. And the first type of prizes I want to mention is actually non-material prizes. And I think they are way more important than material prizes. First of all, we all will accelerate the progress in the longevity field. And again, there are 820 people who registered to fight aging in this hackathon. And it's about 8% of total people who already work on solving aging in the world. So this is really big. Don't get me wrong. What we are doing here is huge. And if we also amplify ourselves with building a lot of AI agents, we will really accelerate the progress. And our parents, our loved ones, our children will live longer lives. What's not to like? Again, second is meet friends, potential co-founders, or just like-minded people. This is always great. People who think alike cannot. What can beat that? You can possibly find jobs or collaborations, and again, in my track, you will have a lot of acceleration. So a lot of VCs already agreed to look at the best teams. Angels already agreed, and I can show you to our partners. So I'll help you, and my colleagues from my track will also help you to accelerate your agent. Okay. With regards to material prizes, in the fundamental track, each challenge gets $3,000 nominated in USDT. There are, I believe, 8 total challenges. And the only one which gets more is, thanks again to our sponsor VitaDAO, is related to female longevity and female health. Because we all love our women, we all love our wives, girlfriends, mothers, grandmothers, daughters. We want them to live long and healthy lives. That's why we are super happy that VitaDAO is also sponsoring our hackathon. With regards to rapid adoption, we have 2 winners who win $2,500 each. Again, the biggest prize comes from VitaDAO because we love our women. VitaDAO will talk later and will tell you more about what they do. Another prize of 2,000 comes from another sponsor, Immortal Dragons, the venture capital firm. Thanks a lot, Boyang. And we have 3 prizes from our computational sponsor, Nebius, and they will come in computational credits. So in total, if you are fast to calculate, there is almost 40K in prizes for you to fight for. So let's have fun and fight for these prizes. And by fun, I mean memes, because we also have a hackathon, which runs alongside the hackathon. And we just extended the deadline until the end of the hackathon as well. So they both end at the same time. So if you wanna, a minute to have some fun, just generate some memes, post them in the hackathon. I give full consent to post memes about me. So please go ahead and have fun. Thank you very much.
Thank you, Alex. Thank you. And thank you for mentioning Nebius, because it's just a smooth switch to our next speaker. It's Suji from Nebius, and I'm really pleased to give him a talk to represent you all the potential of Nebius and this collaboration for longevity future. Suji.
Yes. Thank you. Could I quickly share my screen? That's okay. Yeah. All right. Hopefully you can see my screen. I'll be brief. If you can still see my screen, yeah?
Yeah, we can see it.
Perfect, yeah. So thank you. My name is Suji Maniam. I am a developer advocate for Nebius. So we are really happy to be supporting this hackathon. I figured I will start this with a little icebreaker. I asked the models to tell me a little joke, just to break the ice a little bit, and they came back with this, which is right. I think not too bad. And then I asked another model, tell me another joke, and this is the joke they came up with. So you can be the judge of which one's better, or both of them are equally bad. To me, what's interesting is they both talk about going to therapy. Interesting. So, anywho, Nebius, we have been pretty busy. You may have seen some news. Seen us in the news. It's been a very busy summer for us. Very quickly, we are a global company. We actually provide AI infrastructure. You can run your models, host your models. And another thing we do is we own our entire stack, starting from the hardware all the way to the software that controls it. So what that gives us is pretty good performance and also pretty good value proposition. So we compete on prices. And then the products you will end up using is there are 2 variations. One is Nebius Cloud, and the other one is Nebius Studio. So most of us probably will end up using the Studio because that's where we run our models. And I will talk a little bit about this. And so Studio is we have a nice UI. You can go and you can see all the models we host, and you can run your models. And we host open source models, all the cutting edge open source models we actually host. So we actually have pretty close to 65 models available. You can pick and choose. And just to give you an idea, this summer has been very busy and very good for open source models. So these are the open source models that came this summer. and we have them online. So you have the pick and choose of these are really amazing models. By the way, if you've been following the news, open source models are really good these days. So you can see from the benchmark, these are sort of the closed source models. GPT-5, Grok 4, these are proprietary models. But you can see open source models are very close behind. So the gap is actually very narrow these days. And so we are really happy the open source models are doing really well. And we have all the latest models for you to work on. And there are a lot of things. I'll sort of skip through these. If you look at our benchmarks, we are always the sort of on the top tier for performance, price, and accuracy. So yeah, and I can share the slides with you guys if you like, but I will keep this brief, and I will skip to the slide that really matters. So here's what we hope you guys will take advantage of maybe a studio. We have a credit program here. Hopefully you can take a picture of the slide, and we will share them with you as well in the hackathon area. So if you go to studionevius.com, if you use this code, that will give you pretty good credit. So you can try our models. And if you're looking for examples, we actually have a cookbook available with all the examples. We have lots of agents, a lot of application examples, so you can start from there. And also, if you need some support, we have a Discord server set up as well. And then if you join the Discord using this code, there's a channel that says AI Studio Support. You can post your questions. We monitor them, and we'll be happy to answer. So hopefully you can take a picture of this or take a screenshot and have a great hackathon.
Thank you, Suji. Thank you. And there are a lot of interests from our participants for your slides, so please send us your deck and we will provide the others with it. And just a short change in our schedule. Now I want Daria to have a talk. Daria, are you here?
Yes, I'm here.
Great. Go ahead. Yeah.
Sure. First of all, I really like to congratulate all the organizers and all the participants. I think this is really phenomenal. I wasn't expecting more than 800 people to submit to this hackathon. So this shows that there's tremendous interest on aging, maybe thanks to AI, which for me is probably the most important problem that humanity has to solve. I mean, I cannot imagine anything more important. So I've been very passionate and involved in aging for decades, I would say. I'm an MD, but I've been doing research for the past 35 years. I'm an immunologist. I try to understand the immune system. Obviously, immune aging is extremely important. We try to do some regenerative approaches. But the key question is, can we actually reverse aging? In other words, people talk about healthy aging. Yeah, that's okay, but that really only adds a couple of years to your lifespan. What we really need to think is very big. Bring someone who's in their 70s, 80s back to 20 years old, 30 years old. Add hundreds of years to our lifespans. Of course, this sounded like science fiction to many people, and still does actually. In fact, about 20 years ago, I made some predictions, which were really science fiction back then. I proposed something called biosingularity, which is sort of based on Werner Vij and Ray Kurzweil's singularity idea. And so the idea is that because of the exponential increase in technology, especially computer technology, AI, and of course the biological technologies, that we will come to a point somewhere around 2040s where we will have a complete understanding of the biological systems such that we'll be able to re-engineer them. And re-engineering means re-engineering everything, including aging process. And so bring humans back in life to a younger age. And again, this would only be possible with AI. In that, I've actually had a blog called Biosingularity, which still is there somewhere, where I said that, you know, we will be able to achieve this based on advances in AI technology, because biology is extraordinarily complex. There are trillions of parts and trillions of different processes happening in real time. And so it's impossible for human mind to understand that. But, you know, as we are seeing, and hopefully we'll see in this agentic contest as well, that, you know, with AI, we can really accelerate both understanding of the biology, predicting it, and so on and so forth. And the reason why this is not science fiction and why this is possible, and maybe this is the last thing I want to say, is because biology is not magic, right? It's based on physical principles, and actually it's a sort of a computational system, and it's also very predictable. You know, if you change something, you know, out of a trillion parts, that still has an effect that you can predict. That's why medicines work. That's why biomarkers work, right? So you look at one thing like glucose, you can predict a disease, whereas you have billions of other parts. But more importantly, biological systems are actually self-regenerating and self-repairing systems, right? So our biology constantly renews our body, constantly repairs the systems. Otherwise, you would start to age as soon as you're born. In fact, there's a disease called progeria, where these children, you know, by the age of 8, 9, they become like 100 years old because their biological systems are not able to repair and rejuvenate. And then you have some organisms like whales that can live for hundreds of years, or crabs for maybe 1,000 years. So there's a huge spectrum of how long you can live. And I don't think there is a limit to that, because, you know, we are all in a way immortals. The problem, of course, is that it's very, very complex system. It's a machinery that we don't yet understand, and I'm hoping that, you know, through AI we'll be able to do that. Especially the agentic AI is not only useful for solving the aging problems, but I think in near future it will also be your sort of personal system that will predict if something is going to happen, and so you can really personalize your disease status or your aging based on you know, billions of parameters that the AI systems will collect from you in real time. So it's a super exciting future, and I'm really looking forward to this meeting. And thanks to organizers, Peters, for the invitation. I think it'll be really cool.
Thank you, Daria. Thank you for your talk. I hope you've seen how much support you got for your talk. A lot of people think alike, and a lot of comments. So people really think the same. And I want to give a talk to our next speaker. It's our partner and sponsor, Gero.ai, and Petr Fedichev, who will represent the results of Gero on the field of AI. Go ahead.
Thank you very much for having you, and congratulations on this on this event. It looks huge. So I underreported it on XCOM by suggesting that we have something like less than 700 people. So I represent Gero.ai. So Gero.ai is still a startup. So we use AI to understand biology, to find novel targets against chronic diseases and aging. Our mission is to produce a no bullshit intervention against aging within our lifetime. And I do believe that with AI and machine learning, we can get drugs that work better than eating less. So that's basically the goal of longevity science. We believe that to achieve that, we need a lot of progress and quite non-orthodox views on how to make novel drugs and how to translate them into interventions, including clinical trials and everything else. All of that requires understanding of massive amounts of human clinical and other biological data. And that's why, of course, we need AI to help us to figure out these things a lot faster than we usually do. So agentic AI is a recent advancement on top of the kind of standard machine learning stack. And with the help of some people present here a few months ago, we tried a hackathon for the first time. So we invited a bunch of people from internet to work on one of the problems that my colleague Konstantin will tell you about in a moment. And we were impressed that more than 60 people were working on our problem. And within just a few days, some of them arrived to something that looks like a solution. So we were able to hire these people, and we're proud to be able to work with them. And now, 2+ months after the event, we have something that increasingly looks like a solution. So this will make an impact, we believe, and just in a few seconds you will learn about that. But what I would suggest maybe to finish this already long speech is that, look, at this time, most of us are using agentic AI in order to do things that we can do, right? Just on steroids, on a massive scale. I think the real impact will come from things where agentic AI does things that we cannot do. So I think that all of you, and I suggest that very strongly over these 2 weeks, to think about more problems that can be addressed with agentic AI and actually communicate here with organizers for future events. Because as my mentors in science were teaching me that if you don't have enough imagination, this is really a problem. And lack of resources, of time and everything else, this is just an inconvenience. So really, by having hundreds of people thinking about AI, agentic AI, and aging, I really believe that as one side product of this event, probably we can end up with even better problems than those that are suggested. So once again, congratulations, and please let my colleague Konstantin give you an example of what things like agentic AI can do in real world for biotechnology. Kostya, I've seen you. Yeah, are you coming? Yeah, cool.
Can you hear me?
Yes.
Yeah. No. I think I need to reconnect for a while. Or probably, yeah. No.
Can we?
I can share presentation. Sorry.
I can do it for you. I have your slides. Just give me a second.
No. I think I can share it to everybody. I think it's not a private, so yeah. Can I put it in a chat?
Sure.
Maybe Vita, you would.
Yeah. So I just post it in the chat. Can you open it? Yeah.
Vita, can you share?
Okay. We can switch to slideshow. Yeah. Thanks. As Peter mentioned, it was our first hackathon, and I was a mentor on this hackathon and was quite stressed about whole this event. And I think that's successful team and successful task will be only completed if you work together with your mentors and collaborate with each other. So as much you talk to other people, as better your results you will have. So here is our problem. We would like to use a large language model to extract more data for drug discovery and AI agent. Let's go for the next slide. Next, please. Yeah, so the problematic here is that we would like to find small molecules or drugs which we would like to target our proteins for treatment diseases related to aging or any other diseases, cancer or other diseases. And the problem here is that you have this funnel when you start from a very large amount of input compounds, and you spend a lot of money, thousands of dollars to test it in the lab. And we would like to give this task to outsource to AI models. So, for example, we have some library of potential interesting molecules. We will feed it to AI model, and we'll and the output we will get a few candidates we can use in a clinic for clinical trials. However, this task is very challenging, and I think it's for maybe 30 or 40 years. Nothing exists with a good quality. And on the next slide, maybe we can find the reason why it's happening. Can you switch for the next one? So if you look at the progress in machine learning and artificial intelligence, you will see that very advanced and good quality models have a lot of training parameters. But well, the rule of thumb says that we should have at least 10 times more data points than the parameters. So we are somewhere here where we can find the arrow, and we are very at the bottom on the progress with AI learning. So the best open source dataset contains only 3 million samples. And the situation even worse because if, for example, for imaging, you can augment your data or create synthetic data sets, but for ligand protein data, you cannot do that. So we need a lot of data to train our AI models. Let's switch to the next slide. And can we find more data? Probably yes. So if you go to the largest open source dataset, BindingDB, you can find that about 40% of all data is taken from United States patent data, and it was extracted only from 8,000 documents. And another dataset, short-chamber, it contains 11 millions of these patents with compounds. So potentially there are maybe 1,000 times more data only in US patents, which we can extract. And another evidence that there are more data on this patent bulk is that there are some licensed datasets that are sold for very pricey, and it contains about from 10 to 30 millions records. Let's go to the next slide. So we announced this task for from previous hackathon, and in 2 or 3 days, oh no, it was 1 week, one team, not one, several teams come with a solution when we can use large LLM model agent, not one but two, to extract data. So the idea was that we take data, check this data is good for sending to agent, because we don't want to spend much money. And if agent finds the activity data in these documents, it sends it to another agent which maps activity data for ligands and proteins. After that, we have some normalization, and we get final table. So we started to collaborate with the team Lazarus, which was one of the winners of the previous hackathon. And now we are in 2 months of the progress, and we are going to prepare scientific publication. Let's see what will happen. So let's switch to the next slide. So what our goal is to create a very big data set. So the solution is not ready because you know, on the hackathon you only have approximate solution, and when you go to the real field, there are a lot of problems you have to solve when you need a product ready solution. So from our estimation, we are expecting to have from 5 to 20 millions of data points. So let's go next. So at the current stage, we parsed 6,000 patents from a very short period of time. And we extracted about 50,000 data points, and only 10 of them is known activity data points. So we have 90% of known activities. And for them, for data points which are already known, we have a very good Pearson correlation with existing one. So I think it's quite good results for data which we extracted only by language models, because mostly all this data is extracted manually by people who worked for decades. So for example, these Chambal data sets were collected for 20 years. So and I think it's impressive. Only for 2 months we got these results. So let's go for the next slide. And from my point of view, hackathon is very good for both, I think, for people who are participated in the hackathons and for companies which are sponsored for these hackathons, because we can find talented people and try our ideas. I think that I hope this we will finish this task and publish the results and give a good, how to say, good. Not donation, yeah, maybe the donation for science and longevity. Yeah, thank you for your attention. Do you have any question? Probably I don't have time.
Even if you have, there is no time for the questions. Thank you.
Okay,
Thank you for sharing this. It was extremely important even for me, because I was also an organizer of the last hackathon, and I do love that people joined your team and you did a great work together. It's really very impressive and very actually inspiring for the new participants, I believe. So thank you for sharing. And we continue with our next sponsor. It's Immortality Dragon and Boyan from Immortality Dragons. So Boyan, go ahead. Hi. Boyan? Can you hear me? No?
Yeah, we definitely seen Boyan was there.
Yeah.
Isaac, probably you
Hi.
could start.
Morning, guys. Yeah, due
Morning.
to some reason, Boyan had to go for a while. So I'll take over the intros, and very happy to meet everyone. We got 156 participants in this call. So this is Isaac. I'm from Immortal Dragons. We're a purpose-driven longevity fund based in Singapore. We focus on seeds, pre-seed, bootstrapping companies with radical life extensions and moonshot longevity ideas. We support artificial wombs, whole body replacements, gene therapies. We support those type of companies that are under supported by traditional ventures, right? And besides conventional investments, we also do a lot of evangelism around the world. We sponsor events, we sponsor conferences, we publish books that are longevity related. We connect East and the West, given our deep roots in the East side of the world, especially Asia, on researchers, funds, venture capitals, and also academic resource. things like CROs, top professors from Asian or Chinese universities. And this is our very first actually, participations and contribution to a longevity hackathon. So very happy to see what we can work out by the end of the hackathons, and we're very honored to be supporting the cause. And our website is id.life. Since I'll keep it short, if you have any questions, inquiries, or you want to share your idea with us for potential investments or collaborations, feel free to go to our websites, and there's means of contact that you can reach us and the team. All right. Thank you, everyone, and have a very productive hackathon session.
Thank you, Isaac, and thank you for supporting our rapid adoption track. For the next, being a feminist and being an advocate of femtech, I'm very honored to introduce you Laura from VitaDAO Foundation. Laura, please go ahead.
Hi, thank you so much for having us, and congratulations on organizing this. You obviously have a lot of amazing teams here, partners and speakers. I do have a presentation prepared to explain the challenges. I send it to you. Can you share it, or should I share my screen?
If you don't mind sharing, maybe it's better if you can
Okay,
share.
I'll do that.
Thank you.
Hold on, let me see, make sure that... So I'm going to share my screen. Sorry, you see my dog there. Tools, enter, view, full screen. Hold on. Enter full screen. Is it good? Everybody can see?
Yeah.
Yes.
So I think at DAO, we are the leading decentralized community funding transformative and translational women's health research, education, and funding. And we'd like to say that we're building the field of women's longevity as well. How come I cannot move? Okay, I already did that part. I wanted to tell you about our track record. We've evaluated about now 400 projects across the globe, from scientific labs, universities, and startups. We've done 5 research cohorts, meaning that we've done calls for submissions for translational funding in ovarian longevity, PCOS and endometriosis, gynecological cancers, reproductive technology, and data. We've funded to date over $1 million, 6 labs, 3 preprints, 25 fellowships, and our scientific team for due diligence counts with molecular biologists, clinicians, MDs, neuroscientists, and computational biologists. I don't know why one there. What we do specifically is we do scientific due diligence specifically for women's health, tokenize and fund research with the biggest translational potential, as well as support lab spinouts. We have built an end-to-end ecosystem at the intersection of academia, biotech, and femtech. So just to do a very quick introduction, the problems in reproductive longevity would be, one, it's a really big one, it's data. I know that everybody's very excited about AI, but the algorithm can't compute without data. And when it comes to women's health, we are about 30 years behind. It wasn't until 30 years ago that women were mandated into clinical trials. So again, AI is going to leave women behind. If you can have the most beautiful algorithm, but if you don't have data, it won't happen. The biggest problem is not us also giving you when I wake up, how I feel today, all of those biomarkers, lifestyle we have. What we don't have is tissue data, blood samples, a lot of information that you will need to do this drug discovery. So data is a huge issue. Research itself is also a problem, and that has to do with funding. I'm going to use my favorite meme because a lot of you here are into tech and memes. So our friends at HealthSpan Capital did this amazing research into millions of dollars in funding into the longevity space. So I saw the presentation of everybody being very sad about how longevity doesn't get enough funding. Well, I want to tell you that female-specific longevity gets less than pets. So yay for my dog, bad for the women. And when you consider that women are half of the world's population, it's going to be quite catastrophic if we actually do figure things out. What I want to say is that I don't love problems, I love solutions, and something that is very important that everybody understands. The ovary is a great model to study longevity. Anybody who's in the longevity field knows that Dr. Jennifer Garrison has been speaking about this for a long time. It's not my line. When you think of longitudinal studies, when you think that this is the one organ that dies in the human body within our lifetime, why are more people not actually focusing on the ovary? And part of our mission at VitaDAO is making sure that people pay attention to the things that would actually make a lot of impact. So anybody here who's into longevity and is looking for new areas to work in this field, please think of the ovaries. So our challenge number 1 is quantifying the value. Sorry, I'm just reading off the screen. And I need to quantifying the value of delayed ovarian aging. So collect and analyze available data sets on socioeconomic effects of menopause. And the focus is to understand how delaying menopause could affect productivity, workforce participation, and health costs. Everybody loves to know the numbers. So this is a great challenge because you'll actually be able to come up with very concrete numbers that we can actually talk about when we try to promote more people getting into the field. The second challenge would be personalized models of menopausal milestones, which is during menopausal transition, symptoms appear unevenly and differing really between women. So if we actually figure out a way to get them, analyze them, and input them into other models, it could actually do help in some of these new agenic models. Our challenge prices, we are giving 4,000 in ATH to each one of them. So hopefully with that, some of you can actually get some companies going or join some of the specific projects that we already have that we're working on. And I want to point out that the 2 projects that involve AI for fertility acceleration and menopause that we're working on and that we funded, one of them is Gero from Dr. Peter already spoke. We're very excited. So if you have questions, and since they are part of this, you can even ask them questions. I think it's a very exciting field when you work in AI and also in areas that are overlooked like ovarian aging. And I'm very honored that Peter chose to do this with us. And Life Ahead, Nicholas actually helped put these challenges together. And one of the best known and one of the few researchers that is still in the lab who is an expert at ovarian aging, Dr. Joshua Johnson, is also part of this. So anything that we can help to support during this hackathon to get you the science, information, and everything else, know that you can with the best of the best supporting here and helping you get along. Just as a last minute, I ask everybody if you're curious about the longevity field and also want to get in an area that could be very exciting and massively overlooked, please join us. We have about 35,000 members. We're very active across the globe. I'm very happy that people come and ask us. We're also going to participate in a hackathon in London. I just really want to get more people, even if you're a man, even if you're not that familiar with women's health, into the field of reproductive longevity. With that said, thank you so much and all the best to all the participants. Join our mission and have a great time.
Thank you, Laura. Thank you. It was amazing. I remind you guys that the prize from VitaDAO is the biggest prize for the hackathon. So please join the FemTech challenges. That was really amazing, people say to Laura. Laura, thank you. And we're going to swap again to next speakers. We're going to start with Eric from BIO Protocol to share their new insights on the new Launchpad and AI agents. Eric, are you with us? Yeah, I can see you.
Oh, can you hear me? There we go.
Yes.
All right. Perfect. Well, I appreciate you for having us. Appreciate the opportunity to get to be a speaker. It's nice to go right after Laura, longtime fan of VitaDAO and love the work that y'all are doing. So excited to see some of what can come from that. We've got a couple exciting announcements coming out of BIO Protocol in the near future, which some of them actually have already taken place. So let me just quickly share my screen and see if we can go from there. Let me know if you can see this. Oh, it's going to have me open my system settings. Give me just a second. Zoom is not typically my platform of choice. Let me log back on real quick. I'll be right back.
No problem. No problem. Just to let you know that we want to then to listen to Andrei from Retro.bio, and then we're gonna switch to the challenges, and Andrei will start this part because Retro is also providing us with the new challenge to continue. Yeah, Eric, go ahead.
All right. The host isn't allowing sharing for this meeting. Is there any way you could turn that on real quick?
Do this.
Or if not, I can talk through things as well. That's not a big deal.
Now you're a host. You can do
Awesome.
this.
Thank you so much. Cool. Well, my name is Eric Van Winkle. I am currently functioning as the head of operations for BIO Protocol, and I'm excited at the ability to tell you and the larger group a little bit more about the work that we're doing at BIO Protocol. As a high-level overview, BIO is trying to function as a new liquidity layer for scientific research, trying to enable and fund breakthrough research. For those of you who work in academia, you probably understand the challenges associated with trying to get truly novel and innovative, but often risky work funded through traditional channels, especially in fields like longevity, like women's health. Anything related to some of the teams that BIO has incubated is oftentimes very targeted at groups that have a hard time getting funding. So we recently have started shifting to new models of funding funding scientific research, which are more geared at alignment with the attention economy itself. So in the world of crypto, which is where we come from, a lot of teams will get money by putting, shall we say, a trading fee, a transaction tax on top of information that goes back and forth. So if you can imagine 1% of all of the money that has ever been put through Dogecoin over its lifespan now being redirected towards something positive, that's essentially what we're trying to do. So the website that I'm sharing right now is the launchpad for BIO Protocol, and it shows both the things that we have upcoming, so some of the scientific research projects, some of the decentralized autonomous organizations, and some of the AI agents, in addition to some of the things that we have previously launched. So within this, I want to quickly highlight two very specific things that we've launched recently. The first one being a clone of Dr. Aubrey de Grey. He was generous enough to work with us as a condition of funding parts of the RMR2 study, giving us a lot of the information from his lab so that we can train an agent to be more factual, more accurate, and more precise to the level of rigor that would be needed for an academic. Now, obviously still a work in progress, but we've made pretty incredible strides and are excited to continuously share updates on Twitter about that. The really cool thing about this is that with a 1% transaction tax on the tokens for Gero.ai, we've been able to generate somewhere in the neighborhood of, I think, this says 270,000 right now. Last I heard, it was upwards of 300,000 that have actually been accrued and that can be sent to Dr. de Grey's lab for continued research on RMR2 studies. So really cool things coming out of that. We appreciate his work and his collaboration. Then beyond that, we also see with this agent, a large community of thousands and thousands of people on Twitter who interact with it on a regular basis as a part of the larger incentive structure for the agent, which, believe it or not, has actually yielded thousands of hypotheses. I think the number right now is 2,500 different hypotheses at varying levels of quality from folks who are experts, folks who are not. But there have been some that were good enough that the science team at BIO actually thinks that they should be tokenized and form the basis for future intellectual property. And it's a wonky world in crypto where anyone can make a token, anyone can hold a token, an agent can have its own identity. We dive into some of the finer points of what is an entity on the internet. But the Gero.ai agent actually holds a portion of the tokens from this. So that's a continuous engine building style funding stream that is not reliant on traditional systems. So a part of what we're asking in this particular hackathon challenge is for anyone who's interested to come in and to help us make hypotheses using the Gero.ai agent, such that outputs would be hypotheses which are novel, which would probably have a hard time getting funded by the traditional system, which function as a collaboration between humans and agents, and which there is a viable path to actual experimentation on the hypotheses embedded within. With that being said, I would love to, I guess, potentially, I don't know if we have time for questions, but if not, say thank you.
Thank you, Eric.
We will have a larger session with BIO, so please hold your questions, and we'll be able to ask them in a couple of days.
Awesome, thank you.
And one more way to gain some money, guys. So thank you, Eric. And we're gonna switch to Retro.bio and Andrei. We are very pleased that Retro is supporting us and this hackathon for the second time. Andrei,
Yeah.
please.
Can you see my screen?
Yeah.
Good. Okay, let me. I'm going to introduce our company and the problem in the same slide deck. Thank you for the invitation. So we're going to provide a problem for predicting, protein sequence to function from the basically known papers and databases for longevity. Let me briefly introduce Retro to all of you. Retro.bio is an aging company, aging startup. We were founded 4 years ago in Redwood City, California. We raised $180 million, which is, I believe, the third or the fourth largest aging company in the world. We are about 70 people right now, and our mission is to extend healthy human lifespan by at least 10 years, or preferably more. And for now, we're focusing on 3 therapeutic programs, 2 cell gene therapies, and 1 small molecule program. You may have heard about Retro from our collaboration with OpenAI, which we published 2 months ago. We basically employed GPT-4, OpenAI's best models at that time, to re-engineer some of the factors we are working with, in this case, the Yamanaka factors. And we achieved a very dramatic increase in their efficiency. And you can read more about it, just follow the QR code. The problem that we are going to provide for that hackathon is tangentially related. So we want you to basically democratize that approach and let you also experience the same, awe that we experienced. And the task for you is going to be to think about proteins and their sequences. Amino acids to proteins are what words to human language. And, we know that transformer models and other large language models are great at just deciphering different, connections within the text. And what we did with GPT-4B micro, and what we kind of encourage you to do as well, is to think about those sequence to function relationships for proteins. In this case, you can use basically any protein you can think of. You can pick your own favorite protein, or you can design a more general tool where you can just input any protein of interest for other people to use. But the goal is to try to extract as much information as you can about the relationships between sequence and function. In this case, you can refer to comparative genomics of longevity and evolutionary genomics of longevity, but we don't want to limit your imagination here. So feel free to use any approaches that you find fit for your solution. So in this case, we just show the example of SOX2 and SOX17, where basically it's like you can convert one factor into a reprogramming factor by just modifying a few amino acids within this protein sequence. And that kind of like data we would want you to like try to like work with and try to employ. Maybe to briefly like recap the GPT-4b micro and what we did there, we combined GPT-4o, the basic model of OpenAI that has already learned, it has already read and like memorized multiple papers on like different proteins, their function, biology. It kind of already understands a lot about those concepts. And we added on top of it structure data, sequence data, and coevolution data for basically like all the proteins out there, their interactions and like different protein families. So basically we tried to augment it by additional information to just like give the model more context about, those like papers it was reading, and use it to solve like a practical problem that Retro had. And we basically encourage you to try to do something similar. There are many proteins, there are like 20,000 proteins. You can pick the one that you like, or you think like the one that's the most relevant for aging in your own opinion. And we can just like give you a few hints about like how you can approach it, or how can you can start to work on that. You can use, the Future House WikiCrawl, where they have like a knowledge base about different proteins with descriptions, even though like without like extensive like sequence information about that protein. But you can use it as an inspiration or starter. Think about the sequence of the protein and like linking it to function. The function can be like anything that like, people measured in that specific like database or paper. You can use various aging databases like GeneAge or like any others you can think of for, again, inspiration. Use evolutionary conservation, homolog, similarity across species. If you know that like one species of whales is longer lived, another one is shorter lived, you can, or bats, you can try to link it to the sequence of various proteins that may be responsible for that. And of course, like think about different aging interventions. People tried a ton of mutant strains, knockouts, knock-ins, overexpression, all kinds of genetic interventions into the sequence in, again, like various model organisms, yeast, nematodes, flies, mice, even like human geosis may help here, and human variants. So, and of course, like don't forget about aging, and try to, you can develop like a more general tool, but also you can just like focus on something that's more specific for aging applications. And again, be creative about the coarse graining of those like models or like your approaches. You can focus on specific like, amino acids or, nucleotides, or just like go a level higher to domains or protein families. It's up to you. And again, we don't like want to force you into like very specific like kind of thinking, but do you want to go very broadly, like for like that tool to be applicable genome, like protein wide, or you would focus on like some specific proteins and just like focus on various functions of those proteins and would extensively kind of try to link their sequence to those functions. Would your like database help find an edit, a mutation that we need to introduce to extend lifespan of humans? It's yet unknown. We know that there are longevity variants in the human genomes, so why wouldn't just like we find them? And if you can like provide references or somehow like substantiate your like reasoning, that would be of course like a huge bonus for everyone. I guess I'm ready to take some questions if we have time, or I can just pass.
Andrei was sorry, Vita. Andrei was too humble to say that according to TechCrunch, One of the investors of Retro.bio is none other than Sam Altman. So for many of people on this call know it, but for those who do not, this is something to underline.
Indeed.
Yeah, I am not humble, as you see, so I'm speaking about it.
Guys, and I can just remind you that we're gonna have Q&A session with Andrei tomorrow. So please join the Q&A session for each challenge, and where you can understand the challenge deeper and all the details, and ask any questions regarding to algorithm and data and so on. So Andrei, thank you so much. And again, thank you, Retro, for supporting us. It's real honor for us. And now we're gonna start the next part of our kickoff. It's the presentation about the challenges. Andrei has opened it, and now we're gonna have Professor Peter Litsky from Hong Kong. Thank you for still being with us. I know that's a deep night in Hong Kong right now. But Peter, go ahead.
Yeah, thank you so much for invitation, and this is an exciting session. I'm honored to be part of it. Let me now share my screen. So I work in the City University of Hong Kong. My lab is trying to develop a unifying theory of aging and use it for good. But the task today is broader than gerontology. The belief that science, gerontology itself, and science in general can work much faster than it can work. And the problem why it is a little bit slow is that the way how we communicate knowledge is outdated. So today we communicate knowledge with scientific academic papers. So academic papers emerged like 300 years ago, and it was a great thing for a long time. But now I believe they become outdated. They don't meet the needs and the tempo of today, and they become pure evil. And I will try to convince you why it is so. How does a research paper look today? So it is a novel with the exposure, development, culmination, and final, like 3 to 40 pages long. And this typically describes several experiments. It is quite difficult to read, especially if you are interested in a specific experiment in this paper. You need to go through all these 40 pages to find it. And the research tools to mine data, to search for like specific type of experiments across all the literature emerged only last years. So that with this AI revolution. Before that, finding specific experiments was a non-trivial and time-consuming task. But this is only a small part of the problem. Bigger part of the problem is that each paper is very unique, and it is difficult to evaluate quality and impact. To evaluate that, we need real experts in this particular field to read it very carefully. And this is difficult because you need to organize this session. And with this, the evaluation of scientific merit becomes a very untrivial goal. Alternative way to evaluate research papers is based on citations, how much time this research has been reused. However, currently, each paper contains 50 citations on average, and only 5 to 4 of these reflect the critical evolution of ideas. All the rest are the noise, and this makes citation-based metrics very noisy and uninformative. That's why today, name of the journal that the paper is published is the main criteria for the quality and impact. And this, in turn, creates a huge amount of biases and shortcomings because it creates wrong incentives for researchers. These big papers for big journals takes a lot of time to produce. And during that time, all this data is not visible for the community. Publishing is becoming more and more politicized because very few people decide where, if this paper will be published in this journal. We have reproducibility crisis because scientists are incentivized to oversell their discoveries or exaggerate the effects and to get to these high journals and get careers. So there is also serious crisis that owners of these journals are charging scientists more and more because scientists will pay as their career and grants are depending on this. And now the prices are insane. We have peer review crisis because these few experts that can judge on the particular paper are typically busy and stressed, and they don't want to work for free. And research peer review is now made mostly by non-paid peer reviewers. Many people are saying that the situation is screw up. However, all the KPIs in academia are connected to research papers. So it's very difficult to reform. It is like changing wheels on a moving car. We need first to install new wheels, then move traction to these new wheels, and then remove the old ones, which is very difficult and very interesting task at the same time. So, and that's what we are trying to do. We propose another format, this singularis format. We believe that the basic element of basic citable units should be much smaller than research papers. We believe it should be one logical operation like hypothesis, experiment, method, or dataset, analysis, conclusion. And these elements should be connected with meaningful links, not just with references, which are not annotated right now, but these links should be meaningful. So the idea is that first, we will annotate all the existing research articles with our method and create a graph for each research article. And then we will build connections between the articles using the informed by existing references, connecting individual elements within these articles, creating a coherent knowledge graph that would reflect science in its development. With this, we will produce a tool for efficient human navigation, that it should be much more readable for a human, and also much more accessible for AI-mediated search. Because right now, high-quality scientific searches take about 7 to 8 minutes per request, which makes many high-throughput tasks impossible. This graph will contain links which are annotated and are meaningful. They will reflect real evolution of ideas, and that's why graph topology-based metrics for individual merits will be more accurate than existing ones, like Hirsch factors. And this will probably attract researchers to use these metrics for hiring, granting, awarding. So we also add publication and peer review functionality to our graph at later stages. With this, we plan to make research articles and traditional academic journals obsolete. This is a very ambitious goal, and I don't have enough time to explain it to you in all details. Where we are right now, so now we have an algorithm which can collide the research paper, PDF basically, into this graph connected with meaningful links. So we now have like external facts that are coming from other papers, hypotheses formulated in this paper, methods, experiments, what has been done, results that have been observed, and conclusions made based on these results. So it is working. It is still under construction, but your goal will be to not to improve the algorithm incrementally, but to build an alternative algorithm based on a different technology. Right now, our algorithm runs all LLM. It is time and money consuming and will not suit to annotate 50 million of existing research papers. So we want to build an efficient algorithm based on regular expression analysis and your efficiency will be scored. First of all, how well this regular expression algorithm would fit, will recapitulate the LLM readout. As a secondary criteria, we ask you to produce some meaningful improvement suggestions to the graph structure, like element types, connections, level of granularity, that would serve better the human and machine readability. So, but this secondary criteria will be used only if we have two teams that are really close to each other in terms of core algorithm efficiency. So with this, I want to introduce you to our friendly and fun team. And I don't take any questions.
Exactly. But you know, there are a lot of people who echo everything you said right now. There are a lot of applause and people are supporting everything you told us just now, because everybody faced the same problem, I think. Thank you, Peter. Again, tomorrow we're gonna have a Q&A session with Peter to discuss the task deeply. And now we're gonna move to, thank you, Peter, thank you. And now we're gonna move to Valerie, who is presenting us the task of mammalian phenotype, right?
Correct.
Valerie?
Can you hear me?
Yep.
All right, perfect. And see my shared screen.
Yes.
All right. Well, so yeah, hi everyone. I will describe a project that was created like for this Great Agenda Against Aging Hackathon. I'm like only the voice of this task. Actually, the author is Alexander Ballons, who you contact to via this email. And the project that we are tackling here is simple but fundamental question like, why do we need a phenotype database and how can it truly accelerate progress in like longevity research? And right now, like most of the aging research focuses on genes, but genes like alone don't tell the full story. And what really matters is how genes express themselves, right? So there's like a link, very clear, like very simple biology concept of how genes determines the phenotype. So you are going from gene to trait, like you have some mutations, like small changes in your DNA sequence, and those gene variants are generates like visible difference in phenotype, like a eye color and so on. And for sure, they are not working like that simple. Typically, they organize us into the gene regulatory networks, which are much more complex and still under research. And those are what I need like to justify and remind what the phenotype and genome linkage is. And how we can use it. We know that like even like small changes can be used to create a, like new gene variants and like very different phenotypes. And there is a technology that used to reconstruct extinct like species, those species that are like not on Earth anymore. They were like completely out of that, and they show that how targeted genetic edits can reshape like complex phenotypes, like from the dire wolf to the like woolly mouse. And what about the aging? How can we use it for the aging? Can we use like those techniques or something like that? And for sure, yes, because longevity is like a gene-dependent trait. Actually, our challenge, however, is like different. It's not about creating a new species, right? but about designing genetic and molecular interventions that can shift the mortality curve. Mortality curve is like the opposite curve of the survival curve. So survival curve is like drops from high to low when you consider how some species can dying with the age progression. Mortality curve is like vice versa. So that curve that shows how survival drops with age across the population. And some species, we know it break this rule like drastically. Let's take the naked mole rat. This small wrinkled like rodent lives up to 30 years. It's like 10 times longer than a mouse. And what's remarkable, you can see on this green curve, is that their like risk of death barely increases with ages, with aging. In humans, the chance of like dying roughly doubles every 8 years, like in contrast. And in mole rats, it stays almost constant. They like don't seem to age like in our conventional sense. That's why we mean by shifting the mortality curve, we're trying not to make it infinite, just to make our version of it like look a bit more like theirs, right? Extending the healthy plateau before decline even begins. How to modify the mortality curve is like the unobvious question. We have like a plan. What's the plan is to train a model to predict mortality curves from genomic data, then use that model to generate candidate genetic interventions, then test and validate those predictions experimentally, and then iteratively update the dataset with every result. There's only one problem. There's like no starting dataset exists yet, like not at all. There's like no structural foundation to train the first model. And that's exactly the gap we're trying to fill here in that challenge. Why do we need a dataset? It's obviously the good dataset allows a trained prediction model for gene intervention and then test hypothesis on mice model by creating gene-modified mice. Never find the dataset, so there's like a loop. Reliable data set is like the backbone of any predictive system, we know it. And it allows us to train machine learning models, they can add genes to lifespan outcomes, and so on. And the better the data, the faster the iteration cycle, right? And the closer we get to understand how to intervene in aging. What we know so far, lifespan extension in mice. There is no really existing experiment that would have a radical extension of the life yet. And though we have already dozens of promising interventions, for example, suppressing of some GH, IGF signaling, mTOR inhibition, mitochondrial catalyze, or other genes overexpression, each extending median lifespan by like 10 to 40%. You can see detailed statistics right here. I will not pronounce all of that. There are even cases like partial reprogramming in very old mice, which double their remaining lifespan. And this is a key, no single gene change has produced radical reproducible life extension. There's like no simple parallel between like from the one gene up to like very expensive like extension. That tells us that aging isn't like governed by like one master switch. We need like multi-gene systemic intervention guided by multimodal data. So in that challenge, we propose you to do a pipeline from like to turn literature into structured data, where our goal is to extract like the trait knowledge from scientific papers and transform it into structured, verifiable data sets to use then. This way, instead of like manually reading thousands of papers, machine learning models can start learning patterns directly from the data itself, right? This is like the key point of machine learning models. There is like 3 main stages. First, we search and classify literature by species. Second, we extract the quantitative traits in experimental context, like strain, sex, age, environment, like all the factors that shape phenotype. And third, we normalize and commit everything to provenance-based database. So there's like a very clear trace of where it come from, making the dataset transparent and reproducible. That's also a key point here. About the technical steps, let's start with the step 1. It's like literature search.
I'm sorry, could we switch this part of all the technical details to
All right,
our
sure.
session? For now, maybe just a summary of everything you wanted to say here.
All right, so yeah, I already said all the 3 steps. And as an outcome, we wait a verifiable machine learned ready phenotype data set for aging research, or like longevity research. As an Apple long-term vision, we have like ideas like to develop a self-improving open phenotype database that links genes, trait, and lifespan across species, and a foundation that can empower machine learning models to not only predict, but design and validate iterations. Moving us like one step closer to the data-driven longevity science. That's our main goal, like in the future, based on these developments. And now I can answer any questions if there's any.
No, you can. You will do it tomorrow.
Okay, thank you. Thanks for listening. See you later. Well, sharing. Okay, thank you.
Thank you, Valery. It's quite promising task, actually. We have almost the same in rapid adoption track. So it's really on demand. So please join this challenge. And for now, we're going to have a small video. So it's not online presentation. We're going to see the video which Dima and Simon has prepared for us in advance, because we have a lot of geos from all over the world, so they cannot join us for now. So I will share with you my screen to see the challenge for aging theories. It's also extremely important. I think you can also double this idea, and we're gonna do it just in a second. Sorry. Share. Yep, now you can see it.
Hello guys, I'm Dima from Computage. This short lecture will briefly guide you in our Aging Theories challenge. I will explain you why do we need a theory of aging and how to create a good one. Let's start with some historical remarks. On this slide, you can see how human life expectancy changed across different periods in the past. Before the invention of vaccines, people on average barely lived to 50 years. However, after the introduction of mass vaccination in the middle of 19th century, and later after the discovery of antibiotics in early 20th century, average life expectancy began to rise steadily. And now we've hit a new threshold. You can see how in modern times this curve is saturating. Our new challenge is aging, which we still cannot overcome. What's worse, we cannot even fully understand it. Why is it important to understand aging? Why is it important to have a theory of aging? Let me quickly show you how even a naive but correct theory can be extremely effective. Take immunity. How does it work? It all starts when the body detects an infection. This is done by special immune cells in our body that patrol in real time. Once they detect the infection, then they raise an alarm and call in reinforcements. The arriving cells attack the pathogen using different units against different threats. If they manage to overcome danger, they remember it, so that the next time the pathogen appears, the infection can be dealt with much faster. That's how the immune system trains itself. Now, even from this oversimplified scheme of immunity, you can guess the idea. What if we take a weaker copy of the pathogen and train the immune system to fight it? The result is a vaccine. But more importantly, this allows us to increase human life expectancy by 30 years. Can we repeat this success with aging? So maybe, if only we had a theory. What is the current status of aging theories in science? First, we have the hallmarks of aging, a big list of phenomena observed in aging, such as genomic instability, telomere shortening, mitochondrial dysfunction, and so on. This is a great framework to think about different aspects of aging, but it is not a theory. It doesn't tell us what causes aging, how the different hallmarks are connected, and most importantly, it doesn't suggest any therapy to slow down or reverse aging. A good theory, in contrast, should predict a drug or therapy against aging that we could test in experiments, and then, depending on the results, refine the theory. Searching for an anti-aging drug without a theory looks to me like searching for a needle in an ocean of haystacks. But with a good theory, we could narrow the search to just one haystack, which is still difficult, but at least feasible. Many aging theories have been proposed. Here's a very short list, including some that were suggested almost 100 years ago. They explain aging with varying success at different levels of biological organization, cellular or molecular. Some of them don't even provide mechanistic explanations, but just reduce aging to entropy growth or loss of resilience. Let's consider one of the early theories, the energy consumption hypothesis, which states that organisms possess a finite reserve of some vital substance, which gets depleted over time. Sounds good, but what is this vital substance? Can we measure it? Can we cure aging by replenishing it? This theory gives no answer. On the other hand, there's the DNA damage theory. DNA damage, including mutations, accumulates with age and drives aging phenotypes. This is more advanced than mechanistic theory. It proposes specific molecular change in DNA as the main driver of aging. Accordingly, it points toward therapeutic strategies, such as enhancing DNA repair mechanisms. Unfortunately, even this theory can't explain everything. For example, it purely explains why calorie restriction extends lifespan in many animals. We can also look at the state of aging science from another angle. Here's the top 10 drugs from the drug age database tested in mice. The best one extended lifespan by only about 20%. Not bad, but not a twofold increase. So a great idea would be to build a theory that explains this result and tells us how to improve it. Another recent example shows that a combination of two drugs, rapamycin and trametinib, extends mouse lifespan by 30%. Again, a great idea here. You see where I'm going. Build a theory to explain and improve this result. So let's get to the main point. What makes a good theory of aging? I believe a good theory of aging must satisfy the following 4 criteria. And listen carefully, please, because part of the competition will be based on them. I call such a theory a constructive theory of aging. The 4 criteria are: first, the theory must suggest biomarkers that explain maximum lifespan across different species. Second, the theory must suggest biomarkers that explain mortality within a species. Third, it must predict a testable longevity intervention. And of course, it must be mechanistic, not phenomenological. That means explaining aging in terms of molecular pathways and biochemistry, not abstract terms like vital capacity, entropy, or loss of resilience. Now let's talk about your challenge. In this challenge, you will collect and classify theories of aging. You will be given 2 tasks, which are better done in sequence. We will evaluate your performance to determine the winners, who will receive not only monetary prizes, but also co-authorship in a scientific paper that we will write based on the hackathon results. In the first task, you will need to collect scientific papers relevant to aging theories. The main difficulty here is that not all papers focus on a single theory. Some cover multiple theories. Some may be not about biological aging at all. How to correctly distinguish the relevant ones, that's a job for your agent. As output, you will provide tables in a specific format. More details about the format of tables are in our Discord. In the second task, your goal will be to answer some very important questions from our prepared list. We have also prepared a validation dataset of 10 well-known papers on aging theories, where we have already answered these questions.
Wonderful. While we wait for Vita, I will say that this is probably my favorite challenge in the fundamental track. So if I
might be in another paper. As I said, we will evaluate your results based on the submitted tables and calculate specific metrics. In the first task, the more relevant papers you find, the better. In the second task, correctness of answers is a key. The final score will be based on the average rank of metrics from both tasks. So that's it, guys. I wish you good luck in Aging Hack, and thank you for choosing the Compute.AI challenge on aging theories.
Well, that's it. Thank you, Dima, Simon, and their collaborators, future collaborators, for this important challenge. Because, you know, for the last hackathon, we also tried to hack this theory of aging. And honestly, we didn't succeed enough. So we need to continue and to elaborate on this. And yes, you can get all the slides later. And now I'm pleased to introduce you Vlad from BIO.XYZ, one more task inspired by a company. Someone meant Daronichev in our chat. So Vlad is working with Andrei, and he now is sharing with us his challenge. And Vlad, go ahead, please.
Hello, you're on mute, but we can hear you smiling.
Oh, okay, it now works. Now works. The host didn't allow me to unmute myself. Hi, everybody. So I'm going to present BIO.XYZ challenge for this competition. Please see my screen. So at this competition, basically for this hackathon, we designed a clinical trial outcome prediction task that is done not as it is normally done in the research publications. It's a pretty old problem, but we're tackling it from a very different angle. So first of all, trial outcome prediction task is so important. And it can actually speed up our drug development processes by a lot. Here's an example. In 2011, Derek Love, a very popular medicinal chemist, who's writing a blog post on science, he wrote a skeptical blog post about the Sanofi's drug, which was iniparib, after it failed phase 3 for triple negative breast cancer. And he said that probably, he gave some skepticism and then said, okay, hopefully it will work for other indications. In fact, in 2013, 2 years later, once Sanofi already spent a lot of money, it turns out that iniparib actually fails in all other indications as well. So it failed for squamous cell lung cancer and for platinum-resistant ovarian cancer. And Sanofi basically dropped the development of this drug. And if I would predict that, we would be in a much better shape, and also we could actually oversee that. Sorry, I cannot switch to my screen for some reason. Don't use my Zoom too much. Why I cannot switch the screen? That's pretty interesting. Okay, good. Hopefully you see my slides now. Yeah, they're working. Great. So if we would oversee this before starting the trial, we would first save a lot of money, be more ethical about people who are taking these treatments, and overall, we would probably get to the market some other drug maybe that would actually be more successful. And now we have a sweet spot. We can predict clinical trial outcomes with AI agents, and it is different from what it was from what it has been done in the past, and how people tackled this problem in the past. So in the past, people collected data sets of clinical trials and with their success and failure statuses, but these data sets are kind of leaky, and I will explain that a little bit later. But let's imagine we build an agentic AI system that accesses all the public data with the web tools and predicts not only the binary outcome, what would likely happen, but also gives us reasoning traces of the red, yellow, and green flags of the potential outcomes, which will make the perfect explanation to the reader, let's say a clinician or a medicinal expert, to understand that. And also can do some recommendations on the clinical trial design based on these generated reasonings. So, sorry, I got my slides removed by somebody. Here are the benchmarks that currently being developed for the clinical trial outcome prediction problem, and those all can work in the classical machine learning scenario, but cannot work for the AI agents. And the problem is these benchmarks are basically a dump of the historical of the history of clinical trials from clinicaltrials.gov mainly, and it like covers like dozens of years of past history of clinical trials, and it also means that everything is already in the LLM's weights. So if you would just use current LLMs, they all know about the outcomes of most of these trials. And also, all the data is easily accessible from the web directly, so you can read in the news or wherever else. You will find basically the outcomes of every single endpoint for every single trial, and that's not what we want when designing a proper AI agent. So I'm a big fan of ASCA conference since this year, because I myself joined this conference with a poster. It was in Chicago, and ASCA is an American Society of Clinical Oncology conference. That is huge. So this conference is the biggest conference in the world about oncology. It was about 45,000 people this year in the conference, and it covers a lot of professionals, so medical and clinical oncologists, researchers, sales representatives, and other very, very smart people. And I was pleased to listen to the clinical trial outcomes that were presented for the very first time at this conference. So if you would collect this data from, let's say, every single, not like every single event, but basically, let's just focus on one event of this year, and that will be already substantial and very significant move towards like building a great benchmark for AI agents. And this is what we have done. So we have scraped all the abstracts that are publicly released on the ASCA website, and that was about 6,500 abstracts at the beginning. And that is covering like all the oncology trials of different phases that most of them, not but part of them, were firstly released at this conference. So we don't have, for some of them, we don't have any information that was published before the event that happened this end of May, beginning of June. So if we would take these 6,500 abstracts and then we filter them with all the potential filters, like we need only interventions, and we also need like only drugs, we also need human interventions, and then we can have we also filter for having at least one endpoint with outcome. And most importantly, we need to run some agentic AI system like OpenAI Deep Research to filter out all the data that was published that had any insights about this particular trial in the past before the event of ASCO. So going down for this funnel, we end up with 143 abstracts that are curated, and are ready to be a benchmark and the training data set for our agents. So we also split it like this, and we have some coverage around what types of diseases and indications are there. I will not cover the details right now. You can have a look. And the goal of this competition is to build an agentic system that would not spot direct outcomes that already published in the web, meaning that you are building it now, and probably some results were already published after ASCO conference, right? So after ASCO, there were news about all the trials that were published there. So you probably will find the information easily, but you must restrict your web access to only access the information that was before May 28th of this year, which gives you a lot of room in terms of LLM model selection. So you can pretty much use all modern LLMs because their knowledge cutoff. So because of the matter of how we collect this data set, we will not likely hit any trials that was in the knowledge cutoff of the LLMs, of existing modern LLMs. So you should be free to go with that, but you should restrict the future publications. And if you face any other direct insights about the trials, please also tell us about that, and we might remove that from the data set. So the goal is to build the fair system that really predicts the trial outcome based on the historical data, but not just overfits. And as a evaluation, we are running a Kaggle competition, meaning that you can submit your submissions there as a standard machine learning engineer would do, and you will get the feedback directly. You will see the public score, and later, when the hackathon is done, we will release the private part of the data set. So, I mean, it will be released automatically, and you will see the score of your solution on the private test set. So don't overfit and be careful with your benchmarking and build the right agent. And we can talk about the details of the task tomorrow.
That's right. Thank you, Vlad. Thank you so much. And I would say that it's potentially a quite interesting commercial task, right? Even though it's in fundamental track. Actually, it's also ready for rapid adoption track as well. And yeah, you're gonna answer all the questions tomorrow. And this is a good opportunity for us also to present you another challenge for clinical trials and meta review done by Alex and Albert. Guys, I hope you're here and you're ready to present your task. And thank you everybody who is still with us. We have two more tasks to present. And yep. Alex, Albert.
Yeah, I will share first.
Yeah, hello.
So let me share my screen. You guys can see the screen, right? Great. So thank you everyone for coming. So here, me and Alex, we're presenting you the challenge for the future of evidence. So I'm currently a postdoc in Stanford, working on basic system biology of aging. So long story short, so meta-analysis is a way to integrate various information together to generate a more unbiased view of basically all the results from all the current literature. So it's also been listed as a very top of the evidence hierarchy. So basically, it's been considered as basically the top of actually the gold standard for evidence synthesis. So it's right above the randomized control trial. And but why it matter for aging? So it's quite straightforward. So aging is very heterogeneous process, and many lifespan studies, especially in mice, are usually done by different lab with a relatively small cohort. So you might see paper very frequently that some compound or some genetic modification could extend lifespan, but sometimes not replicate well in other lab because lifespan is a very highly heterogeneous trait. It's being affected by various factors like the lab environment, how people handle the mice, or how do they give the treatment, etc., etc. So it's become more and more important to gather all the information about the intervention of interest, and then aggregate all the evidence, and then give us a final view of whether this intervention will indeed change the lifespan of mice, and how much does it change. So I will just quickly give you an overview how usually it's been done by a human researcher, and then Alex will introduce you how AI agent could help it. So usually it's called systematic review and meta-analysis. So it's have a standard pipeline. It's called Pickle. So basically, you first define your population, which is if it's animal study, whether you focus on mouse, or whether if you focus on certain strain of mouse, or whether a mouse with certain genetic mutation, and then what the intervention you'll be interested in, and what comparison are you looking at, and then what outcome are you looking at. And after you define all these problems, all these questions, and then you will have a preset criteria for the literature selection. And after that, you write down your search strategy. Like I will use what keyword and on what database to search all the relevant literature. And after you gather all the literature, the next step would be after you make up a plan to search for literature, and that next step would be really searching the literature and select what study you need to include. Like you will need to choose the major database for paper, and then you do the literature search just given a keyword, and you need to decide whether to include a preprint or not, or other kind of report kind of paper. And then you based on the quality, but before you do that, you need to have a certain rule, like what quality are you looking at, what sample size, or what kind of study you need to filter out. And then you have a set of the literature that you think is highly relevant to what you are interested in. And the final step would be just a statistical analysis. So basically you extract all the statistics from the paper, usually effect size and p-value and sample size and the error term. For example, lifespan extension would be either the hazard of mortality or the real lifespan number and the error bar and statistics. And after you get the statistics, you can perform some very standard meta-analysis. Like you can use fixed effect model, random effect model to harmonize all the effects together and eventually give you a final output. So this is one example from previous literature. So basically what you did is you're looking at several previous intervention paper, and you may notice that it's not significantly changed the lifespan in majority of paper, but after you met all the information together, the result give you more accurate estimate. Basically, it's turned out that this intervention can indeed change the effect, and it has a very significant effect. So all this will help you basically ensemble the information from the scattered literature and find the intervention really help with aging or lifespan. So Alex will introduce more later. So maybe Alex, you can jump in. You're muted.
Oh, thank you, Albert. Yeah, so I'll present you our challenge and what we expect from you, what we expect from the AI agent that you built. So, first of all, what is the current problem we have? We have only 10,000 aging researchers in the world, and there are so many clinical trials out there already published that it's just impossible to analyze everything by such a small community. And to help us, we are now using AI. And so our challenge is to build an AI agent that will automate meta-analysis of clinical trials. And there are actually over 500,000 clinical trials already published. So even if you have a large team, it's impossible to go through all of that. That's why we need AI in this case. So the AI agent that you will build should completely automate the meta-analysis. It should prepare a nice report. And also you have to optimize model efficiency so it doesn't burn too much money. So just a little bit about the workflow. Just as Albert just told you, we should automate each step a human is doing. And actually, we already know all of the steps because they're pretty standard. First, you have to search the literature. Then you have to select the relevant clinical trials based on the quality, inclusion, exclusion criteria, and so on. That's also very standardized, and you can use AI agents for that. Then from the studies that you find, you have to extract the statistical data, tables, effect sizes, and so on. And finally, you just use the standard statistical analysis of these clinical trials, which is also very standard. And as you see, this problem is actually not that fantastical. It just you have to build the whole pipeline yourself using AI agents. And we will evaluate AI agents based on the following 4 criteria. So you have to select studies accurately, you have to extract data accurately, your agent should give valid statistical results, and also your agent has to be time efficient, so you don't burn too many resources to make a clinical trial. Still, it has to be more efficient than if it was done by a human. And we prepared 2 already published meta-analysis that you will be tested against. So we'll talk about them next days when we discuss the everything in detail. Also, we will provide you all of the help you will need with clinical trials and with large language models. So we have like essential reading on the how clinical trials are done. We have all of the resources for accessing the clinical trials themselves, API and so on. And also we will provide you help with large language models, which models to choose for data extraction and so on. So that's it. And let's change the future of evidence together. Thank you.
Thank you, guys. Thank you for the presentation and a good work on. Yeah, you see a lot of emotions sent to you. People are amazed by your challenge. Great, thank you, guys. And we're gonna switch to the last one. Maybe you mentioned that we missed Nick with the Femtech challenge. It's just a technical thing. We're gonna come to you tomorrow to explain the Femtech challenge. And now it's Alex from Axiom to tell us about his challenge on toxicity. Alex, are you with us?
Yeah, hey there. How's it going? Thank you so much for the invite. I really appreciate it. It's really fun to be part of this. Axiom is a company that works to build AI tools to help scientists assess drug toxicity more accurately and more affordably than physical experiments. So we're working with a bunch of big pharma, and we've raised about 40 million from CRV, Dimension Capital, Amplify, and Jeff Dean. And on average, each new approved drug saves, the best data I could find is in the UK, and each new approved drug saves about 20,000 quality-adjusted life years in the UK alone. Probably saves millions of quality-adjusted life years worldwide. So from a certain perspective, drug discovery is actually succeeding at extending lifespans. It's just happening much, much, much more slowly than we'd like. And we'd like to make this process much faster. And liver toxicity is the biggest cause of failure in clinical trials. The main problem with getting new drugs to market is the fact that of the hundreds of clinical trials that we start each year, and of the thousands of drug programs that we start each year, only about 50 drugs will make it to the market after many of them fail due to efficacy or due to toxicity in clinical trials. About 25% of those failures are liver toxicity. And there was 1 week last year where companies lost a combined $5 billion in market cap due to liver toxicity related failures. So it's a huge problem, both scientifically and medically and commercially. And one of the reasons for this is that existing lab experiments are terrible at predicting clinical outcomes. So benchmarks from big pharma across the board show that existing in vitro experiments and in vivo experiments have poor sensitivity and specificity at predicting clinical outcomes, while still being hugely expensive. And so why don't we use AI to help address this problem? It's an extremely hard problem because the right data sets don't exist yet. Molecular space is incredibly large and diverse. So if we want to train something that predicts toxicity from small molecule structure, we need really big data sets. But existing lab experiment data sets tend to be quite small. And usually, if they're data sets that have already been created that folks are exploring and using, they usually only include very simple endpoints, like did some cells die when you added a drug? And then existing clinical data sets often run only into the small hundreds of drugs. They often don't include dosage information. And there's a lot of sampling bias and distribution shift over the years. So we need to make the data that we need to solve this problem. And this is what this challenge is going to be about. Why you might want to participate in this challenge. Well, liver tox is a massive problem. And for each clinical trial failure that we avoid due to liver tox, we can save hundreds of thousands of quality adjusted life years if this results in a drug making it to market that wouldn't have otherwise. We're also a small, fast growing startup. We're trying to grow a really awesome talent dense team. And if you do super well in this challenge, then we'll want to talk with you. So what will you do? We'd like you to start with the FDA DILI-rank dataset and use this to test your solutions. And then we also have an internal benchmark set that we've created from clinical trial literature, which we'll keep hidden, but we'll use to evaluate solutions. And you can do one or both of two things. The first is to focus on clinical data and build AI agents to create clinical toxicity outcomes from the web. So you might want to design an agent that can look up different drugs and classify them as toxic, non-toxic, or unclear, and also create the dosage information for those drugs. Or if you're a bit more ambitious, you might want to create an agent that can create specific frequencies and severities of different liver toxicity outcomes for each drug, maybe pharmacokinetics information as well. And there's a lot of information about these sorts of things around on the web, including clinicaltrials.gov, papers, patents, press releases. Another option is to focus on preclinical or lab experiment data. And here you could either build agents or use your own biological or data-driven insights to collect relevant data and figure out whether it's useful for predicting clinical outcomes. And there's a long list of different databases and data sources that you might be interested in considering for this, including simple assays, including RNA sequencing data, binding data, and more. And then the ultimate goal is to train models across any of the data that you create to predict drug-induced liver injury outcomes. And you can use this FDA DILI-rank data set as your both training and validation set to start with. So the deliverables, we'd like you to provide a cross-validation ensemble of models. The splits should be generated with scaffold split, and we'll link you to a library that will do this for you. This ensures that the similar molecules are not in training and test. And each ensemble member needs to record the IDs or scaffolds of the molecules that are saw during train time. Then we'd like you to provide an inference function that we can call, which takes in a drug's representation and its dose and predicts the probability of toxicity at that dose. So the important thing here is to ensure a fair evaluation for your ensemble. Only the ensemble members that were not trained on a particular drug should be allowed to assist in producing predictions for that drug. So it's out of cross-validation fold predictions. And then also code to reproduce the data collection and model training. So the way that we're going to evaluate this is quite simple at the end of the day. We're going to compute an ROC-AUC score between your predicted drug-induced liver injury probabilities and the labels on our test set. We're only going to compute that AUC over the portion of the curve where false positive rate is under 0.5 or specificity is over 0.5. There's a flag for this in scikit-learn, so it's super easy to run. But what this essentially does is it only computes ROC-AUC in the high specificity area of the curve. So you should focus on making a small to moderate number of high confidence toxicity predictions and predicting lower scores for anything that's not high confidence. We'll try to help out. I'll shortly provide an example eval harness, which will call your function to evaluate a model on DILI-rank in the same way that we'll evaluate it on our internal test set. And I'll also be available on Discord to answer questions as you go. So yeah, thanks so much, and we'll be around to help out as things go on. Vita, do you want to do questions now, or we want to roll on?
Thanks a lot.
We're gonna
Vita,
do
go
it
ahead.
asynchronic through Discord, okay?
Cool, perfect.
Thank you, Alex. I know that you are busy, and thank you for sharing your time with us today. Thanks. Thank you. And guys, Alex, you wanted to tell something? No? I want to tell you that we finished with the fundamental track. And now before switching to rapid adoption track, I do want to introduce you and to give a talk to a person who truly needs no introduction for anyone from the longevity field. It's Aubrey de Grey, who really honored us to join this hackathon kickoff for today. So Aubrey, please tell us your ideas regarding the longevity and AI agents.
And how does it feel to be an agent? Before, in the beginning, we were told that there is an agent of yours. Welcome.
Right. So, so first of all, thank you so much to all of you, to the team who've organized this, for telling me about it and for having me along. I've really enjoyed the last 2 hours. I had to step out during Peter Felichev's talk, but I've been able to listen to more or less everything else. And I'm absolutely delighted at how successful this initiative appears to be, and will be. I do not have the details of the timing for the discussion tomorrow, and I'm going to do my very best to make time for it. So if somebody could let me know about that, that would be much appreciated. But yes, a few remarks. Well, so yes, some of you will know that I used to actually work as an AI researcher 30 years ago before I switched fields. And some people who do know that seem to assume that that means I know a lot about AI, but it's actually not true. AI research back then was completely different from what it is now. So I don't really feel very knowledgeable about the area. But of course, I am absolutely astounded and overjoyed at the magnitude of the contributions that AI is making in this space at the moment. When I say this space, of course, I don't only mean longevity specifically. Advances that are more general in their applicability are just as important. The most obvious one, of course, being AlphaFold and its successor AlphaProteo, which I believe is going to be enormously valuable. I think it has yet to really reach its potential. But in terms of longevity and regenerative medicine, the enormous advance that was made recently by Retro.bio in collaboration with OpenAI, which we heard about earlier in the session, that's it blows my mind. Most especially because of the sheer number of amino acids that were changed by the agent, by the LLM. Literally more than half of them, if I remember rightly. That's something that experimental techniques such as directed evolution would never be able to achieve. So we have to definitely understand that we are in a completely new world in terms of being able to adapt existing amino acid sequences for improved function. And in fact, I'm already in discussion with Joe Betts about another application for that, which I very much hope we're going to be able to get off the ground very soon. But I'll talk about that later. Other than that, I think I really want to just talk about the hackathon, because it's just so impressive, not only that you have so many people here, but that the quality of the ideas, the challenges that have just been presented is so enormously good. I would like to, actually reflect on a lot of those and on the contributions that they may make as time goes on. I'd love to know more about the hackathon as it progresses. I think I'll just mention one suggestion for the moment, which is in relation to the challenge concerning theories of aging. I think there's a case for expanding it a little bit. Some of you may know that I actually am not a fan of the idea that there are generic mechanistic theories of aging that we have failed to discover at this point. If we talk about the entire animal kingdom, I already felt that 20-odd years ago. I felt that we had already reached diminishing returns from what we could learn about human aging by studying nematodes. At this point, I think we've reached that point with mammals. I think that I think it's very unlikely that we will ever discover any new mechanistic theory of aging that is truly generic across the whole of the tree of mammalian life, and that there's plenty we need to discover and that will be valuable for us as we develop biomedical interventions, but that those things will be organ-specific or species-specific. They will not be generic. So I would like, since the challenge, as I understood it, was defined in terms of seeking precisely that, a generic mechanistic theory of aging that we haven't yet discovered, I would like the challenge to be expanded so that it allows someone to be rewarded for developing a negative result. In other words, for developing new and concrete persuasive evidence that there is no such theory out there waiting to be discovered, because that's certainly the outcome that I very much expect to occur. And I think there may be a little bit too much emphasis in the community right now on the idea that, well, we haven't done much about aging for a long time. We haven't made much progress in mouse lifespan, for example. Therefore, we must have a deficient theoretical framework, and we just need to improve on that as the next step. I think that's completely wrong. I think that the thing that we must remember is that the body is trying not to age. The body is always on our side when it comes to staying healthy. And the only reason we actually suffer functional decline late in life is because the body's tools to do that are not completely sufficient. So our job is to augment those tools. And in many cases, that augmentation can be done with very primitive tools. In fact, the presenter on the theories challenge, I think, mentioned the early days of immunology and the development of very primitive methods for augmenting our immune resistance to infections, which saved many, many millions of lives. I believe we need to go in that direction. And a large part of the reason I believe that is because we do now have a wide diversity of ways to extend mouse lifespan a little bit, including many that are not or seem not to be calorie restriction mimetics. That's a category that has received far too much attention in the gerontology field over the past 30 years, especially. And the reason it's far too much is because we already know that it will not scale to long-lived species in such as humans. So I believe that so long as we focus on the fact that we have a diverse range of non-CR mimetic interventions that are already somewhat successful in mice, we don't need another theory. We just need to focus on what I'm calling the unknown knowns in gerontology. Of course, we all know what known unknowns and unknown unknowns are. I believe that this is a case of unknown knowns where we've got a bunch of things that we already know about and we just haven't combined them in an effective way. So that's, of course, the focus of my work at LEV Foundation. I believe it also needs to be very seriously considered by the community at large. So I'll stop there for the moment, and I will look forward to tomorrow's session where I gather, lots of people are going to be able to answer questions. I'm sure there will be a lot of questions to all of the people who presented today. I just want to say again, I've had an enormously good time over the past couple of hours listening to all of you, and I massively congratulate everyone involved on pulling this off. Congratulations and thank you.
Thanks a lot, Aubrey. Now, those of you who haven't met Aubrey, I don't know if these people exist, but you understand why we needed to make an AI agent to amplify what Aubrey can do to the field, right? So Aubrey can be everywhere right now and increase the speed at which we iterate and move forward. So that's it from the speakers for the fundamental track and special guest Aubrey. So there will be one speaker, it is me. I will tell you more about the rapid adoption track, and then we'll have some time for Q&A. So hello again. You have seen me in the beginning. I'm Alexei. I was actually introduced to the field and I would say indoctrinated by Aubrey de Grey, the previous speaker. So I applaud him for bringing me to the field. So I try to do my best to accelerate the progress so all of us live longer, healthier lives. And that's what is the purpose of our rapid adoption track as well. It is different from previous problems because we provide you with more freedom to actually choose what to work on. So this is for those who want to switch their careers to the field, for entrepreneurs who want to find a good challenge to solve in the nearest couple of years. So the ideal scenario, what will result from this track, would be 1 or 2 companies getting started. I wouldn't be happier if this would be the result of what we are doing here. Also, you've seen that there is an actual launch pod for AI agents. So there are new ways to finance AI agents and the development teams. So we will talk more about it in the nearest future. We will explore how to do that. And if this is the route you want to go, this is also available. And again, I've prepared for you a lot of VCs and angels and other partners from basically everyone in the longevity field. So I will help you with introduction. I will help you with crafting your message in a way that it will actually help them solve some problems, and otherwise accelerate you so we all fight aging in a better way. So basically, about the high-level purpose of this rapid adoption track is to accelerate the advancement of the longevity industry with agentic AI. And there are 4 ways to accelerate the industry. First, it is raising awareness. And you would be surprised how little people care about living longer. So you have to, ask a lot of questions, and you are met with what we call death acceptance propaganda a lot. So people just, they have their crisis, they realize they are mortal, they cannot do anything with it, and they develop all sorts of ways to actually, okay, I will die, and that would be it. Death is okay. What about overpopulation and whatever? So we need to actually raise awareness amongst the general public that actually doing something with aging is not only possible, it is being done already, and we need to be doing much, much more. Another big bottleneck is funding. There are some creative ways to attract funding. Again, BIO protocol, several DAOs, a lot of venture capitals. Actually, at the same time, we started one of our partners, Longevity, the venture capital firm. They announced they are launching Fund 2. So congratulations to them. And one of the representatives of the fund will actually help you with the acceleration. So we will see how to actually break into the venture capital with longevity. The third bottleneck is actually attracting more people into the field. And I've talked about it. Alex talked about it 15 minutes ago, that there are less than 10,000 people working on aging. This is, madness for me. I don't know, like 20% of world population should be working on it, because people are dying because of aging. So we need to attract everyone, like from politicians, scientists and engineers to designers, various sorts of creative people, anyone. So like there are, this is a meteor coming to us and like killing 100,000 people tomorrow. Another meteor and killing 100,000 people the day after tomorrow. And we're not doing enough to prevent it. And the fourth bottleneck that is a part of the high-level purpose of this track is actually generating or unlocking more data, and that is what, for instance, Peter and Konstantin told about in the beginning, how they were able to generate a lot of new data from patents which were just lying there, and this data was never tapped. So there are vast reservoirs of such data which can be actually generated to accelerate the field. So how it is done? So we have about 7 challenges. They are loosely defined. And so you can take one of these challenges and you can actually modify them. You can use them for the inspiration, and then use some modified challenge which would serve high-level purpose that I was mentioning before. Or you can actually suggest your own challenge. We think people are very smart, and you probably know better, and we actually seek for inspiration and ideas from you. and you can suggest your own challenge to solve and actually hack during the hackathon. So this here in this QR code or in this link, there is a little bit more information about these challenges. This is how this agent might look like. How we would evaluate the challenges. So again, it is either a business project or a social impact project. Both are very important, and I would argue the social impact is even more important now because how much of an acceleration it could provide. So we either assess you from the business point of view or social impact point of view. Then the 30 points is given to the quality of demo. So it's like how well the code does run, how clear documentation is, and how engaging a demo video which you would record would be. Other 20 points is given for longevity impact, and another 20 points for agentic fit. So whether agentic AI is a good tool to use here. The decision will be made by judges, physical people who will just see each project and give scores to each project. Then we will choose the winner. I will talk more about it later. Again, a little bit about confirmed VCs and angels who would join us. Immortal Dragons you've seen, VitaDAO you've seen, Longevity I told you about, 2060 Foundation is a group of angels, and some people will join, but they asked to remain in stealth mode, and these are very impactful people in the United States, I can tell you that. I really hope we'll be able to announce this during this hackathon, but it will be huge collaboration in the next hackathon if anything goes right. So fingers crossed. Immortal Dragons and several other people will join. Again, some people reached out to me and they need engineers as well. They said, okay, just show me one or two best engineers. So like it's a great idea to do your best. Even if you don't win, like if you do good work, there are non-material benefits to this hackathon. Again, I already told you the price structure. There are two winners. Each receives 2,500 USDT. There will be a special prize by VitaDAO, which is the largest prize. So there is an incentive to choose a project in female longevity or female health. There is a special prize by Immortal Dragons and 3 special prizes by Nibius. And here is important information I mentioned before. All these special prizes will be decided by the sponsors themselves. So they have our own way to decide who will be the winner. So that it's basically their own decision who they will like the most. Again, so if you want to be a part of the Rapid Adoption Challenge, here is my schedule. Please scan it. I will also put this link in the Discord, so you can have a 20-minute talk with me where we will think about your challenge when we actually meet each other, and where I will have the opportunity to help you to shape your challenge and probably find the best mentors, the best potential collaborators, and whatever. So like this is a very good opportunity. So I cleared out 3 days and sometime on Saturday. So in the beginning of the hackathon, you will have a lot of opportunities to talk to me to actually decide on what to build. And yeah, that is it. I will leave it for a little bit more time so everyone could scan it or photo it. But again, I will put it in the Discord.